You're coming home from school one evening and realize that you might be coming down with a cold. Your family urges you to get some rest and you'll be fine, but when you wake the next morning you feel much worse. You skip school to see the doctor, but by 10 a.m. you're nauseous, lethargic, dizzy, and pale. It's winter, but you're dripping in sweat. You finally arrive at the doctor's office, only to be ordered to the hospital for an IV and a myriad of blood tests. Your body has been expelling fluids for days, so your veins are collapsed. Finally, the nurse finds a vein. You get the fluids, but you're still motionlessly laying in the hospital. The blood tests come back the next day. There's a bacterial infection in your blood, but the lab couldn't identify the strain. You accidentally overhear the doctor informing your nurse to just pump fluids and antibiotics into you, to just keep doing something, despite the fact that nothing has relieved the sickness. You lie in bed for one more day. You feel your body rise one more degree. You let your nurse try one more antibiotic. After that, the antibiotics stop coming, but your infection worsens and your temperature continues to rise. Antibiotics represent a cornerstone of modern medicine. For over 80 years, we've had the ability to fight the entity associated with disease, infection, and plague. Since the introduction of antibiotics in the 1920s, we've had the ability to effectively kill the microscopic organisms that brought the bubonic plagues to our ancestors and killed millions of people. However, this is slowly changing. We're starting to see that bacteria evolve resistance much faster than we can create new drugs. Are we losing the arms race with bacteria? The first question is, why is this happening? How are bacteria becoming resistant? Some bacteria have genes that make them resistant to our antibiotics. So when a bacterial population is subjected to antibiotics, these individuals survive and continue to reproduce. Now the problem is, if you're always killing the non-resistant bacteria, the resistant ones are the only ones left. A population of bacteria that was once nearly eliminated by a dose of antibiotics comes back, and now they can no longer be treated with the same antibiotics. And these resistant bacteria can even share their resistance. The genes that grant resistance generally aren't stuck in the genome, they are mobile. The resistance genes can be replicated and sent out of the cell as plasmids and be taken into other bacteria. So, every time bacteria are subjected to antibiotics, it adds selective pressure for antibiotic resistance. One study in Europe looked at the correlation between antibiotic use and resistance in different countries. The study found that countries that used penicillin more had more bacteria that were resistant to the drug. It's been seen in this figure that France had both the highest usage and resistance. In addition, antibiotics are often overused or misused. For example, only about 10% of antibiotics that are used in agriculture to keep livestock healthy are actually used to fight off infectious diseases. Instead, they are used prophylactically or to promote growth. And the antibiotics are used at much higher doses than are necessary. This leads to increases in resistant strains of bacteria that can move through the environment and impact human health. The development of new antibiotics cannot keep up with the emergence of these resistant bacteria. So what can we do? Instead of developing more antibiotics and adding to the drugs we both knowingly and unknowingly pump into our bodies, one idea proposed by Ojala and his colleagues was to find a way to discourage bacteria from sharing their plasmids, so that instead bacteria would cease to share anything at all because it was bad for them. There are these special viruses meant for bacteria called bacteriophages. It was found that certain bacteriophages are able to screen out and destroy bacteria containing antibiotic-resistant plasmids. Ojala performed an experiment where he infected bacteria with a bacteriophage, and he discovered there was a significant decrease in the number of antibiotic-resistant bacteria. Some of the bacteria had stopped sharing their plasmids. In this figure from the study, the blue boxes represent the number of resistant bacteria without phages, and the yellow boxes represent the number of resistant bacteria with phages. As you can see, with the addition of bacteriophages, the levels of antibiotic-resistant bacteria decreased by several orders of magnitude. Even in the presence of antibiotics, bacteriophages managed to discourage the bacteria from becoming resistant. By day 3, the phages were still able to lower the number of resistant bacteria. In these cases, the bacteriophages were successful. Now let's talk about the lower right-hand box. In this case, the bacteria were treated with a double dose of antibiotics. For the first day, the phages successfully decreased the amount of resistant bacteria. But by day 2, the phages were no longer able to discourage bacteria from sharing their resistance. With the extra pressure of 2 antibiotic treatments, the bacteria were more interested in protecting themselves through plasmid sharing than not. In this case, the phages had lost the fight. An important point to understand is that whether we're using antibiotics or bacteriophages, there will always be a battle with drug resistance. The method proposed by Ojala and his colleagues may not necessarily be the solution to our problem. However, it represents a new strategy for a seemingly endless battle. Using the mechanisms and applications of evolutionary biology, we will have a better chance at fighting antibiotic resistance.